Good morning. Welcome to the grand rounds of the Family Medicine Department at Mount Sinai School of Medicine. My name is Eric Gale. I'm the President and CEO of the Institute for Family Health. This morning, we'll be hearing about emerging genetic diagnostic approaches to undiagnosed diseases from researchers at the Mount Sinai Center for Undiagnosed Diseases. The center is part of a national undiagnosed disease network project funded by the National Institutes of Health, and the Institute is a partner in the Mount Sinai Center. Patients of all ages with difficult to diagnose diseases, including Institute patients, can apply to have their cases studied intensively using state-of-the-art genomic approaches. We are engaged in this work to ensure that the Undiagnosed Disease Network project capabilities are made available equitably. Myself and Dr. Saskia Schumann, the Institute's Assistant Vice President of Research Operations, serve as community investigators on the project. Together, the team is working to provide this unique resource to patients in the New York metropolitan region and to engage in communities underrepresented in medical research with the ultimate goal of identifying rare diseases and helping patients access the care and information they need. We're pleased to welcome Dr. Carol Horowitz, Louise Bier, and Rachel Evard to speak with us about this. Dr. Carol Horowitz is the Professor of Population Health Science and Policy and Professor of Medicine at the Icahn School of Medicine. She's the founding director of Mount Sinai's Institute for Health Equity Research and the founding dean for career advancement and support. Dr. Horowitz has been a key research partner of the Institute and the Department of Family Medicine, and we have a long history of collaboration, including on previous genetic studies, such as the APOL1 study. Louise Bier is the Director of Clinical Trials in the Department of Genetics and Genomic Sciences and Director of Clinical Research in the Genetic Counseling Training Program at the Icahn School of Medicine. Rachel Evard serves as a research genetic counselor and site coordinator at the Center for Undiagnosed Diseases. Louise and Rachel are both certified genetic counselor with master's degrees in genetic counseling from the Icahn School of Medicine. Please welcome this team, and I hand over to Dr. Carol Horowitz.
Good morning, everybody. I apologize in advance for community engaged research, and it's always been an honor to partner with the folks at Institute for Family Health. And when I had an opportunity to get involved in genetics research, the first people I reached out to were Neil Kalman, Eric Gale, Diane Hauser. And when we started this, a lot of primary care providers, including folks at IFH, had a healthy earned skepticism. Our patients have so much going on. Why are we doing genetics? But what we all learned is our patients wanted it. And as Neil said at that time, we want our patients to be the first, not the last, to benefit from advances in science and medicine. So we've had a many years long partnership. And when this opportunity came to bring a chance for us to say, which one of our patients do we think something's wrong and we can't figure it out? It's really complicated and something else is going on. The first thing we thought of was working with you all on this. I will say that my daughter has a couple of rare diseases. Because of the curiosity, the holism, and the understanding of complexity.
Great. Thank you, Dr. Horowitz. Good morning, everybody. Lovely to meet you all. So let me get my slides shared. Bear with me one moment. Get in presenter mode. Wonderful. So thank you, Dr. Horowitz and Dr. Gale, for your introduction. I'm very happy to be here presenting on behalf of a whole team at Mount Sinai and IFH that's working as part of the Center for Undiagnosed Diseases. I'll be talking today a bit about some emerging genetic diagnostic approaches. I will try not to nerd out too hard or too long on any one specific technology. Interrupt me if I start to do so. All right, we have no conflicts of interest to disclose. So the learning objectives for today are first to identify the uses and limitations of exome and genome sequencing, which are being used clinically as standard of care in many cases, to understand new technological approaches that can help to arrive at clinical genetic diagnoses for patients, types of technologies beyond exome and genome sequencing. Third, to identify appropriate patients for referral to the Mount Sinai Center for Undiagnosed Diseases. And fourth, to explain the clinical, diagnostic, and support resources that are available at the center. Just before I get too deep into the weeds on genetics, I did want to give you just a small short overview of the Undiagnosed Diseases Network. This is a research study that is funded by the NIH, and it brings together clinical and research experts from across the country to solve the most challenging medical mysteries using some of these advanced technologies that I'll talk about today. The program goals and objectives for the UDN are first and foremost to improve the level of diagnosis and care for patients with undiagnosed diseases. Second, to facilitate research into the etiology of undiagnosed diseases. So to better understand what types of causes might be underlying some of these very difficult to diagnose cases. And third, to create an integrated and collaborative research community to identify improved options for optimal patient management. So not just stopping at diagnosis, but really seeing how can we then better care for these patients in the long term. So I am going to take a step back and talk a little bit about Genetics 101, just to kind of lay some foundation, remind us all of some of our previous biology classes that we've all taken in the past, but to make sure that we're all on the same page with some of the basic concepts that will help to understand some of the technologies I'll be talking about. So we're going back to the central dogma. So this is really kind of the foundational concept of how the genes and the DNA that are in the nucleus of each of our cells, how that ultimately gets translated into the proteins that are the building blocks for our bodies and for all of the processes and enzymes that are needed to make our bodies run. So we start with the DNA, which contains both introns and exons. The exons are considered the coding portions of our DNA. These are the parts that instruct our cells how to create proteins. The introns used to be considered kind of junk DNA, just filler, but that's actually not really true. There's actually a lot of complex information contained in the introns, and those might be things like regulatory elements, so things that tell a gene when to turn on or off, or portions that instruct how much a gene is expressed or how little a gene is expressed. So there's important things happening in the introns, even if they're not directly coding, giving the instructions for how to create a protein. So we start with the DNA, those bases, those introns and exons, and then through the process of transcription and splicing, we get to RNA. And you can see here that RNA consists of just the exon portions. The introns have been removed through a process called splicing. And then from the RNA, we have translation, which basically then creates the protein. Forgive me, my cat is yowling in the background, if you can hear her. You'll see here one important thing to note is that at this point of transcription from DNA to RNA, there are actually a few different paths that can happen. There's a concept called alternative splicing. So not every exon may be included in every piece of RNA. So here, if we were to follow this little diagram on the left, the gene has spliced in exons 1, 2, and 4, and we get this bubbly cloud-shaped protein. And on the right, the gene has spliced in exons 2, 3, and 4, and we get this smooth circular protein. So this idea is basically that a single gene based on different modes of splicing can actually lead to different types of proteins. Okay, so that's the central dogma. If everything works perfectly, fantastic. But what happens if there is variation or changes in the starting DNA in our cells? So it's important to remember that on average, each of us carry millions of variants in our own DNA. This is normal. Variation in genes is actually what makes each of us unique, one compared to the other. And so if any of us were to be sequenced right now at this minute, if we were to compare our sequence to the standard reference genome, we would find millions and millions of changes. Not for the vast majority of these changes would not cause clinical symptoms. But there are instances where a change might be pathogenic and might be considered disease-causing, where it could lead to a severe type of genetic condition. So just over on the right here, I'm not going to go into too much detail on these diagrams, but you can see a few different examples of types of genetic variations. We have the normal reference genome at the top, and then down below you can see an example of a substitution change, and then insertion and deletions. Variants, depending on their impact on clinical disease, can be classified on a scale from benign, which is not having any clinical impact, to pathogenic, which is causing disease. One quick note is that in the past, we often referred to these changes as mutations. And that was kind of, you might still hear that kind of used interchangeably to mean pathogenic variants, but more and more we are referring to them as pathogenic variants, really just as a recognition of the fact that variation itself is normal and occurs in everybody. Okay, so next I'm going to talk a little bit more about a few different genetic tests that are being used right now in standard clinical genetics care and in a research setting through the UDN that can be used to detect variation in patients where we think there might be an underlying genetic cause. So I'm going to talk specifically here about exome and genome sequencing. I will say there are many more types of genetic tests that are being used clinically. This is not an exhaustive list by any means, but these are kind of the most advanced tests that are being used and used relatively commonly. So whole genome sequencing and whole exome sequencing are quite similar. They are both reading the DNA at the level of the base pair, meaning the A, T's, C's, and G's that make up our genes. Whole genome sequencing, as it implies, sequences everything, introns and exons, coding and non-coding regions. Whole exome sequencing focuses only on the exons, so only on those portions that directly code for the RNA that then leads to the protein. Excuse me, the coding portion of our genome really only makes up 1 to 2% of our entire genome. So whole exome sequencing, because there's less material involved, actually can be a more cost-effective genetic test. So after sequencing, after detecting and reading through each of the individual bases, finding the variants, we then actually have to interpret those variants and try to understand what type of impact they might be having clinically, whether there's any clinical relevance to those changes that we've seen in particular, and interpret those variants on that scale from benign or likely benign to pathogenic or likely pathogenic. And you can imagine, as I said, if there's millions of variants in each individual person, that this is a very complex and intensive process. In some ways, the technological sequencing itself is the I shouldn't say the simple part. Everyone who works in a lab and who works very hard to make sequencing happen would not agree with that. But I will say that interpreting the sequence variants is as much of a challenge as the technology itself. The interpretation of variants, there are standard clinical guidelines that are followed across labs. So there is consistency from lab to lab. And these labs consider a handful of different criteria, different pieces and types of evidence to try to determine whether a single change is going to be disease causing or not. I'm going to talk through these in a little bit more detail. So the first criteria is population data. So we have a number of resources, databases that have sequence data for tens of thousands and hundreds of thousands of individuals who are, by and large, for the most part, generally healthy, meaning without severe early onset disease. They're not necessarily patients that we can say have lived an entire lifetime without any significant disease, but they don't have, a severe early onset presentation. So if we have these large databases of relatively healthy individuals, we can take a variant of interest and look in those databases to say, well, do we see the same change in any of these ostensibly healthy people? If you see that variant in a relatively high frequency or relatively common proportion of those healthy databases, then you can say, okay, this is probably unlikely to be causing a severe genetic presentation in my patient. On the other hand, if it's never been seen in any of those patients, in any of those databases, that might cause you to kind of keep it on your list of potential causes, potentially being disease causing. So that is the population criteria, population data evidence that we can use. Second is computational and predictive data. So there are multiple and very advanced different types of computer models that will basically try to predict what happens when you have a change in a DNA, what that does to the RNA and the ultimate protein, and what that might mean for how that protein interacts with all of the other proteins that it's meant to interact with. These computer models are not perfect by any means. They cannot capture the entire complexity of our human biology, but they can be a piece of information. They can consider things like, okay, does this gene change cause a polar amino acid to become a non-polar amino acid? Or does this change occur in the binding portion of a protein? Or does it occur kind of at the tail end that's not really an active region of the protein? So those types of pieces of information can be fed into these computer models, and the computer model can give a prediction. So again, not going to be a silver bullet to tell us, what is the correct answer for any particular variant, but another piece of evidence that can be considered. A third type of evidence is functional data. This is basically taking a variant and doing an experiment to see in a cell model or in an animal model to see what is the impact of this variant in these cells or in this animal. Again, this is not a perfect silver bullet way to understand the impact of a variant. These models, again, are not replicas of human beings, but they can serve as another piece of evidence. I'll talk a little bit more about some functional studies later, so we'll get back to that. Next, we have something called segregation data, and this really has to do with family history and how the condition might be presenting in a family overall. As family medicine practitioners, you can appreciate, I'm sure, how important the family history is for a variety of reasons, not just for purposes of genetics, but it warms my heart to know that I'm talking to a group of people who likewise care deeply about what an extensive family history might consist of for our patients and how meaningful that might be. So with segregation data, what we are looking to see, if we're looking to see, okay, does the disease that we're evaluating our patient for occur in other family members? If so, we can then look at those family members and see, do they have this same variant of interest? Does this variant occur in the affected family members? And likewise, does it not occur in unaffected family members? If we see that the variant is tracking or segregating with the actual disease presentation in the family, that's another piece of evidence that this variant might actually be the cause for the condition we're considering. And then finally, are pieces of evidence around whether a variant is de novo, whether it's occurring for the first time, and allelic data. So that's really getting at whether or not the variant is inherited directly from parents or whether it's occurring anew for the first time in the patient. De novo variants that occur for the first time in the patient, that in and of itself is somewhat unusual, and so can be considered a piece of evidence that this variant might be pathogenic. Great. So whole exome and whole genome sequencing are amazing. They are very powerful types of technologies, but they're not perfect. They have limitations. One of the limitations is around intronic variants. So first off, whole exome sequencing is only looking at exons. So obviously it is missing those deep intronic variants altogether, simply because the technology is not looking for them. Whole genome sequencing, even though it captures those intronic variants, the interpretation of what they mean is a challenge. We're pretty good at understanding how variants in exons might impact a protein ultimately, but we're less good at understanding the various regulatory elements and recognizing what they might mean, changes in those regulatory elements might mean. So if a change is occurring in a portion that switches a gene on or off, that's just really hard for us to recognize from the base reads itself. A second limitation is around identification of structural variants. So just from a technological standpoint, whole exome and whole genome sequencing are not great at picking up larger changes in genes. They're great at reading down to that single base pair level, letter by letter, through the DNA, but they have a harder time recognizing, is there a whole chunk of DNA that switched between two places? Or if there's a stretch of DNA that has a repeated sequence of bases over and over and over, it has a harder time reading through that stretch and understanding how many repeats there might be. And then the final limitation is really just due to the fact that we are still limited in our knowledge of how the complexity of genes, RNA, and proteins work, and understanding what that means for human biology. There's a lot we still don't know. So we're often, even with all of those pieces of evidence that I talked about previously, population data, segregation data, computer models, there are still many times where we have a variant and we say we just don't have enough information to know whether it's benign or pathogenic. And so it stays in this category called variants of uncertain significance. Likewise, there are still many genes where we don't know exactly what the role is of that gene in human biology, or what might happen if that gene is not working exactly properly. And so in those instances, we might also find a variant Not only is the variant uncertain, but the meaning of the gene itself that it occurs in is uncertain. So we can also have genes of uncertain significance. So there's still a lot we're learning, which inherently is a limitation of our ability to fully interpret all of the data that can be generated. So next, I'm going to talk a little bit about some of the new types of genomic technologies that can be used to help us fill in some of these gaps and limitations in exome and genome sequencing. Excuse me. So first, I'm going to talk about RNA sequencing. So if you remember our central dogma, DNA to RNA to protein, whole exome and whole genome sequencing are reading through at that top level. They're reading through the DNA itself. RNA sequencing is reading the next level in that, which is the RNA. So it is reading through after splicing has occurred. And RNA sequencing is a method for sequencing the entire set of RNA molecules, which you also might hear to referred as the transcriptome, because it happens after transcription. It reads through all of the RNA molecules in a tissue sample. And this can be used to help interpret variants of uncertain significance. It's important to know that RNA-seq results will be different tissue to tissue because different tissues need to express different genes to fulfill their different functions. A muscle cell is not expressing the same genes. It doesn't need to do the same thing that a brain cell does. And so here is just a little kind of simple diagram showing that idea. Here on the left with this green neuron, we can see that gene 3 is very highly expressed and gene 1 is moderately expressed with a little bit of gene 2 being expressed. On the right, you see the muscle cell, which has a moderate amount of gene 3 and gene 4, a little bit of gene 1, so different from the neuron itself. So RNA sequencing is really helpful because it can give us two important pieces of information. First, it can detect changes in the RNA sequence itself, giving us information on what the final protein chain will be. So this means that we can better understand if a variant impacts how the introns are spliced out in that step going from DNA to RNA. So over on the right, it's a little bit small, but you can see a series of little diagrams that show different examples of splicing that might occur. A variant might cause an exon to be skipped altogether. In that second line, you can see the brown exon is just skipped incorrectly, or it might cause an intron to be retained, or different types of issues caused with splicing. So that's one way that RNA-seq can be helpful, but it can also tell us how much a gene is expressed. So rather than looking at the RNA sequence itself, we can also see whether there is more or less of a gene being expressed compared to normal, compared to what we would expect. And so here on the left in that little graph, you can see an example. That little red dot way up in the upper left is showing an outlier indicating that, okay, this particular gene is much lower, much less expressed than what we would have expected. So RNA-seq in combination with exome and genome can actually increase the diagnostic yield of those tests by 10 to 35% compared to doing exome or genome alone. So it's a really powerful tool when used in combination with exome or genome sequencing. I'm going to go through a case example here. So this is a case from the UDN, one of the other UDN sites at Baylor College of Medicine. And they had two siblings that they were evaluating. And they had a clinical diagnosis just based on their symptom presentation of something called Fine-Lubinsky syndrome. And this is characterized by craniofacial anomalies, hearing loss, cataracts, microstomia, which is small mouth, CNS anomalies, and developmental delay. This syndrome, based just on this unique clinical picture, was first described back in 1983. So physicians were able to diagnose patients with this condition based on their clinical picture alone, but it was over 40 years until we actually understood what the underlying genetic cause of this syndrome was. So Baylor in 2024, just to give you a little brief background, these patients did have prior genetic testing. So they had a couple of different types of tests that both were uninformative. When they were referred to the UDN reanalyzed the exome data that had been generated and tested prior, and they found intronic variants in a gene called DPH1. So these are variants occurring in the intron that would have been too difficult to be able to say with certainty whether they're benign or pathogenic. And so Baylor decided to do RNA sequencing analysis to see if they would be able to better interpret these variants of interest. And here is what they found from their RNA sequencing. So the blue, green, and brown lines on the bottom are the controls, and you can see there where the bars are high, that's an exon that's being expressed. Where you have the little string, that is the intron that's being skipped over to the next exon that then gets expressed again. One of the patients is shown in red up at the top, and we can see two things of interest here. So first, at that red arrow, that is where the intron typically starts in the controls. And you can see that sometimes that's where the patient's intron starts. But then at the yellow arrow, you can see that they actually have a little bit of gene expression that extends into the intron, indicating that that's being spliced too far along. So the intron is being included when it shouldn't be. So we see incorrect splicing. And then the other thing that is, I'm sure you all have already noticed, is that the red bars are much shorter compared to the controls. So we're also seeing that the gene, even with the incorrect splicing, the gene is just not being as expressed as highly as it is in the controls. So we're seeing both incorrect splicing and abnormal lowered expression, which are good pieces of evidence that the variant that they're working up is a good candidate for being disease-causing for these two siblings. Great. So next I'm going to talk about another type of technology called long-read genome sequencing, which can help to fill the gap in detecting larger structural variants from exome and genome sequencing. So I realize you might be saying, you've already told us about genome sequencing. What's this difference with long-read? How is it different? The main difference is in the length of the read. So when you sequence DNA, the DNA basically gets chopped up into bits and then fed through the sequencer. Those lengths, those bits are fed through, and each bit is considered a read. In traditional exome and genome sequencing, it reads about somewhere from 50 to 300 bases at a time. So those are relatively short reads. Long read genome sequencing chops the DNA up into much longer pieces, anywhere from 1,000 to 20,000 plus bases. So much longer stretches of DNA are being read in one go. And this is what allows for better detection of those large deletions, insertions, duplications, rearrangements. And it also allows for detection of those repeat expansions that can happen in those highly repetitive areas. And this is a very simple diagram to try to illustrate this a bit. So on the top, you can see, I went that way. Okay, on the top you can see an example of what a short read might look like. Again, in a very simplified way. We haven't put hundreds and thousands of variants here, but you can see in this example, we have a read that says GGA, GGA, GGA. The reference genome down here, there is a long stretch of that pattern, and this little short sequence could fit in multiple different areas of that pattern. And it's impossible to say, is this sequence belong here at the second instance of the pattern, or does it belong at the last instance of the pattern? We don't know. A long read is going to be long enough that it actually has the anchor points on either side of that stretch of repeats. That way we know exactly where this read matches. It's a little bit, to think of it a little bit like if you had a work of Shakespeare and it was cut up into individual words, and you had a word the, and you had to find where it belongs in Julius Caesar. You are not going to be able to know where that single the belongs in the play. But if you have an entire sentence, then you actually have more information. You know where it matches, where it should fit in within that broader piece. So again, another example of how long-read genome sequencing has been used in the UDN to make a diagnosis. So this is a case of a 61-year-old woman who was evaluated at Vanderbilt University. She had adult onset epilepsy and cortical tremor. Her first seizure was at 38 years old, and it was stimulus sensitive. She had a family history of adult onset seizures and progressive movement disorder, primarily on her father's side of the family. And from a clinical presentation, this is associated or consistent with familial adult myoclonic epilepsy. This condition is associated with multiple different genes. There are many different genes that can cause this presentation. So she had prior genetic testing, trying to find out which gene might be the cause here, but all of that prior testing was negative, not informative. And so she then had long-read genome sequencing through Vanderbilt and through the UDN. And they found one of those repeat expansions. So by doing long read, they were able to accurately count the repeats occurring in a region that were then considered pathogenic. So typical, there's the repeat here, it's a 5 base pair element of repeated 4 T's followed by an A. Typically, most people have 20 or fewer instances of that repeat. On her first allele, so on one of her genes, you can see that she has only 15, so that's normal. But on the second allele, she has over 600 repeats of that stretch. And that then is considered to be pathogenic, and a diagnosis then was made for her. Vanderbilt was able also to work with the rest of her family and her other family members that are affected. And you can see here, the circle with the arrow is the patient, the primary patient. And then you can see her affected brother, her affected father. These are the ages of onset, not their ages at evaluation. And you also see an affected female cousin on her father's side. And the very much too small little red bars are just showing that the repeat was observed in all of the affected relatives as well. Her uncle, who had the same condition, was deceased, and so he was not able to be tested. But I think this is a really good example of how the diagnosis made through the UDN actually then has impacts for the rest of the family. So the last type of technology I'll talk about are animal models, and this gets at the functional studies. So if we have a variant or gene of uncertain significance, animal studies are one of the types of experiments that can be done to see what is the impact of that variant or gene on another organism. There are a few different kind of general types of models. So you might hear the term knockout study, and this is where a gene is made entirely inactive in the model. Then there's knockdown, where the gene is made partially inactive. The activity is reduced, but not eliminated altogether. And then there's knock-in, which is basically inserting a variant or a gene sequence to see what the effect is. And there are all different types of animals that can be used. Mice, flies, zebrafish, they might have different benefits or issues depending on the specific context, but lots of different animals can be used for functional studies. So for this case example, this is through UCLA and Baylor College of Medicine, who are both involved in the UDN as well. And first, an institution had a patient, a 6-year-old girl, who had growth and developmental delay. They observed a variant in this gene called SRRM1. The gene, this was a suspicious variant for them, but they did not know anything about SRRM1 yet. They didn't know what it did. It's a gene of uncertain significance. So it has certain elements of evidence that makes it suspicious, but they don't know enough yet to say anything with confidence. So the team submitted this case to a site called GeneMatcher, and it's going to sound silly, but it's basically like online dating to learn if you have genes of interest that you're interested in, a way to match up with other researchers who are also interested in the same genes. So you submit your case, you say, hey, I'm interested in meeting anyone else who is also working on SRRM1. And through GeneMatcher, they actually connected with 2 other institutions around the world who each had an individual patient. You can see here, one is an 11-year-old girl, the other is a 17-year-old boy. The clinical presentations are not exactly the same, but they all kind of have a bit of a neurodevelopmental, possibly a growth-related presentation. So we now have 3 patients, and they wanted to learn more about what does this gene actually do. So they created a knockdown fly model. So they actually had 2 different colonies of flies. One had gene expression reduced by 50%, one reduced by 30%. And they did a couple of different tests on these flies to see if there was a neurological impact. So one is called the Bang sensitivity test. And not to get too in the weeds, I think it's actually kind of interesting, but basically you expose the flies to some type of stimulus and they basically have a brief spasm and will kind of lay prone for a fraction of a second, and then they will resume their normal fly-like behaviors. And you can see here that in both the knockdown one, so the first knockdown colony, and the second knockdown colony, it took longer for the flies to resume their normal behavior compared to the controls. They then did a second type of test, which is annoyingly called negative geotaxis, which just means climbing. So they basically, flies naturally climb upwards. You shake the flies down to the bottom of your little tube where they're housed in and see how long it takes for them to climb up the sides. And again, in both of the knockdown models, you can see it took significantly longer for the flies to climb up the sides compared to the controls. So we have these couple pieces of evidence that, okay, at least in flies, reducing the activity of this gene does seem to have some type of neurological impact. So this is a good example where, the process started by trying to identify patients, a group of patients, and learn about them together, and then to use these functional models to generate additional evidence for this gene being a likely cause for the neurodevelopmental disorder that we're seeing. Great. So now that I've talked a bit about the different types of technologies that can be used in the UDN, I'm going to talk a little bit more about the network itself. So this was, as I said, initiated at the NIH in 2008, and in 2024, Mount Sinai and Institute for Family Health in partnership were awarded a grant for phase 3 of the study. The phase 3 priorities are to develop plans for long-term program sustainability. This is a really valuable resource, and we want to make sure that it can be implemented for years and years to come, not simply reliant on the NIH. And then second, to make sure that we are recruiting people who have been previously underrepresented in the UDN. It will surprise no one on this call to know that the UDN, as with many research studies, has been quite homogeneous in the patients that have been enrolled. And so we really want to address that in this current phase of the project. There are sites all across the country. You heard me mention them. There are clinical sites, there are research sites, there are sequencing cores. And these are a couple of the specialty cores where some of those specialty assays, special tests that I talked about can be performed. There are other types of tests and approaches that can also be used. So again, this wasn't an exhaustive list, but just kind of to give you a little bit of a taste of what can be done. And as I said before, this really is a team effort. So we have a number of people involved, both at Mount Sinai and at Institute for Family Health. I know Dr. Gale kindly introduced us before. He is a co-PI at IFH, as is Dr. Saskia Schumann. And then Dr. Julie Schoonover is the clinical champion and co-investigator at IFH, which we are so happy about. This really does take a lot of people to make this work. I'm running a little short on time, so I'm going to skip over this slide. I want to talk about the inclusion and exclusion criteria for the program. So who should be referred for evaluation in this program? Patients who have one or more objective findings and who don't have any diagnosis, even though they should have been, kind of worked up by relevant specialists already. We can enroll patients of any age, children, adults who don't have the capacity to consent for themselves. Obviously, we would work with their family member or guardian, and we can also enroll in any language as well. The ideal, I hate to say ideal UDN participant, but the patients where we're kind of most likely to be able to make a diagnosis are those who have multiple body systems affected, an earlier age of disease onset. If there's relevant family members available, that's helpful, but none of this is required. And then just to give you an example of a few different types of participants that are not types, a few different actual participants that are involved in the program, and these are all shared with participant permission, of course. You can see here it goes across a range of ages and a range of presentations. So, we really are looking for you to think outside the box. We're not looking for any one kind of cookie cutter type of patient to be referred to us. And actually, I'm going to turn it over to Rachel Evard, who's going to talk a bit more about the case submission process.
Great. Thank you so much, Louise. Next slide, please. So now I'll talk a little bit more about how your patients can actually make it to our center. So our team's created a REDCap eligibility inquiry where providers can submit a de-identified case summary, similarly to the example shown here on the right. These inquiries should include, the patient's medical concerns, including which providers or specialists have evaluated them for the condition, and any pertinent evaluations such as imaging, lab work, and genetic testing. I also want to note that we are developing a referral mechanism that exists directly within Epic through the CareEver referral management pipeline for IFH providers to just be able to submit this inquiry directly through Epic. We expect to begin working on this in Q1 of next year, and IFH providers will be alerted that the mechanism has gone live through Epic Infos. I also want to emphasize that the eligibility inquiry can absolutely be used for cases where you're not completely sure if they meet inclusion criteria, right? This is a new study. We're all still trying to learn which kinds of patients might most benefit from this study. Overall, we want this to be a really collaborative experience, and our team, might even reach out with additional questions about the patient. In the case that the patient isn't appropriate for formal submission to the study, but might benefit from a clinical genetics evaluation, we would then help to facilitate a warm handoff with our clinical genetics team at Mount Sinai. So please don't worry about submitting any cases to our team, even if you're unsure if they're appropriate for this study. Next slide, please. One more. Yeah. And then in the case that the patient is appropriate for further evaluation in the study, we would then provide next steps for formal case submission and support the patient in that process. One thing to note for referring providers is that a recommendation letter is required, and we do have templates that are available, and we can absolutely provide them to you at that time. And then I would support the patient in gathering any medical records that are required for formal review of the case. One thing to note is we do have an authorization form that the patient can sign, which just allows our team to review records that are directly available in Epic. This will hopefully minimize the burden of, patients gathering their own records. Next, please. And then within 60 days of receiving all that requested medical information, our team will make a decision and inform the participant and the referring provider of that decision. And then the UDN evaluation would take place. Now I'll talk briefly about the different types of case decisions. And of note, the UDN uses a tiered process. So every case that comes to our team will always receive a thorough medical record review, but cases assigned to Tier 1 will only receive this medical record review. Tier 1 is really most appropriate, as seen on the slide here, for patients where a diagnosis was identified upon the review, maybe a potential beneficial referral was identified, or the participant might just be more appropriate for some expert site outside of the network. And we always provide next steps recommendations for our Tier 1 participants. And then individuals assigned to tiers 2 through 4 will receive an actual UDN evaluation, with tier 2 participants receiving a telehealth one, tier 3 receiving in-person, and tier 4 having some research component, such as those animal models that Louise had described earlier. Our enrollment goal across the 4-year grant period, which originally started in August 2024, is about 200 participants. So I want to emphasize that overall it's a relatively small program. And so this isn't really somewhere where your referrals might get lost or you'd never hear from our team again. And then in terms of our program billing structure, for sustainability purposes, and because many of these clinical evaluations and the majority of imaging or testing that's ordered is just routine clinical care, any participants that have accepted insurance would just follow standard billing procedures, with the exception that any research tests wouldn't be billed to the participant. I also want to note that we have a patient assistance program through the National Organization for Rare Diseases that can also help to assist participants with out-of-pocket costs and travel. And then for our uninsured participants, we will use grant funding and philanthropy to cover any of those evaluations. And I want to emphasize also that our team, never takes insurance into account when making case decisions or reviewing the cases. And then now that we've given, quite a broad overview of new emerging genetic technologies available through the network, I want to talk a little bit more about how this program could help IFH patients specifically. And I know Dr. Gale and Dr. Horowitz already mentioned previously, but Mount Sinai and IFH have a really long-standing relationship. And we really hope that this close partnership with IFH through the network will continue to strengthen these existing relationships and also elevate care for our underserved patients. We also hope that the program will provide greater access to research initiatives and emerging technologies in genetics to IFH patients. And lastly, as I mentioned before, it's a relatively small program in the grand scheme of things. And so we really aim to continuously improve coordination and follow up with all of the referring providers of the program. And I wanted to conclude and reiterate a quote that Dr. Horowitz had told us earlier today by Dr. Neil Kalman, who's the co-founder and former CEO of IFH, which our team felt was really insightful and valuable in the context of this program, which is our patients should be the first, not the last, to benefit from advances in science and medicine. We are really eager to make this program available and accessible to the patients at IFH. And with that, we hope to receive case submissions from you all so that we can ensure that the IFH patients can also benefit from the available resources of the program. And then on this last slide, we just have some contact information for myself as well as the center. Louise, next slide. And please never hesitate to reach out with any questions that you may have about the program or any patients that might benefit from the program. Thank you so much for listening. I know we don't have so much time left, but Dr. Horowitz, let us know if you have anything else that you'd like to emphasize or mention, and happy to open the floor to any questions as well at this point.
No, it's a pleasure to be together. And as a primary busy PCP myself, I know it's really hard to imagine prioritizing this on top of all the other things we do to manage our patients. But I do already have in mind one of my patients where we just can't figure things out, and they think this could be a really rare and wonderful opportunity for us to provide care for our patients we've never would have gotten to before. And IFH is the only that I know of FQHC in the entire network, and I know we're going to crush it. We're going to show everybody why this partnership matters and why it's meaningful.
Thank you, team. I do think this will help us identify those patients, because I do believe we're seeing these patients in our clinics, and I think the challenge is identifying that they could benefit from this referral. So one of the questions I had was, if you're familiar with you're taking care of a patient, they seem to be low risk, but you have enough family history to suggest that they may be at risk for a particular condition. But an individual patient you're seeing is risk appears to be low. Is that a patient that you should still be recommending for an assessment to Undiagnosed network?
So it sounds like in this situation, the patient themselves are not presenting with symptoms, but they have a family history that you're concerned about. Is that right? Yeah. So in this situation, I don't think that would be right for the UDN, because we're really with the UDN looking to work up the patient who's presenting with the symptoms. But that does sound like they might be a good referral for our general clinical genetics team to meet with. As Rachel said, you can basically send us your cases and we are happy to connect them as needed. So we work within clinical genetics. We work with these providers very closely. So we're happy to make that connection or to provide that type of kind of feedback on any individual case that you have.
Thank you. Let's see if there's any questions. See the screen here.
I don't think I see any questions in the Q&A, but happy to take any questions, any follow-up questions that people might have now or in the future. Are there any treatments once you identify the variants? So that is really kind of the next frontier in genetics and genetic care. It really depends on the condition that's diagnosed. Unfortunately, well, it truly depends. There are day by day more and more treatments that are being tested for genetic conditions. There's not necessarily a lot. So it depends. With the UDN, we're making really kind of one-off diagnoses. So we don't know exactly what we might find for any particular patient. And there might be, in some cases, there might be treatment recommendations or guidelines that might result from a diagnosis, but it's hard to predict. Wonderful.
Great.
Thank you so much. We will be happy to send around our contact information and the link to submit case inquiries. Again, we are happy to don't be shy about submitting cases. We're happy to think it through with you directly. And thank you all so much for your time this morning.
Thank you, Carol. Thank you, Louise. Thank you, Rachel.